Thanks, Dr. Raghavan, for this opportunity and the Indian Academy of Sciences for this opportunity. I'm a fellow from 2023. This is my first talk in this kind of conference. So basically, because I know there is a varied expertise here, so I'll keep it, this is a complex study, but I'll try to keep it under control. So I will talk inside the cells. So everything is inside the cells of our body. So please keep that in mind. So my lab work on cell biology, and in cell biology also we are more interested in innate immune responses of our body and other aspects like autophagy and other, which I'm not going to talk today. I'm going to talk about one of our recent work published, which is programmed cell revival. Many of you have heard about programmed cell death during your, maybe in bachelor's or before bachelor's. Programmed cell death is, what is it? It is most of the cells which are dying in our body are very well programmed, means they are dying by a programmed process. And this is very well established from years that how they actually go towards the death. And programmed cell death is very important in every day in our body. Millions of cells might be dying by programmed cell death, and it is important to maintain, to keep us away from many diseases, including cancer and autoimmunity. So what is this programmed cell revival? Okay, so I would like to convince in like 10-15 minutes that what doesn't kill you will make you probably more stronger, at least at the cell level. Okay, so this paper is published in AMBO journal, and this was present on the cover where you can see the phoenix rising, and that's what it depicts from our study. So this study was not actually distant. It was totally observation-based study and totally a serendipitous observation. We were studying something totally different. We were studying the cell death, actually. And this guy, Kautilya, he was my first PhD student, and he wanted to understand the cell death, some aspects of cell death, and specifically lysosomal cell death. That's, if you trigger the lysosomes, you know all lysosomes probably, there's a part of cells which is like a suicidal bag. If they burst, they will kill the cell. So here it was, these are all agents which can kill the cell by lysosomal burst. Okay, so I'm keeping it all these things simpler. So what he saw is, what he was trying to see is how lysosomal cell death happens. What happened is, he went to the lab, he started this experiment to understand. So what happens is, this is when you give these agents, the cells get these morphology, means they are towards, all started blabbing, and they come out of the surface, which is the property of these cells. These are fibroblast cells. They come out of the cells into the media. And this is the point where this guy has to collect the cells, because we want to study at this stage. Luckily, sometimes these things are luckily, he forgot it to collect these cells. And when he went after around 1 hour, because he forgot it, this come first. These cells are like, standard apoptotic cells in literature. And most of the people at these concentration collects these cells. There are hundreds of papers on LLOME. So they all collect these cells at these stages. So you can see that this is faster, so I will go with this. So they completely get detached, they have all apoptotic things going on, and then they revive back. Okay, so those also revive back, or different magnification these cells are. Okay, so we try to understand, so what we did is, we try to understand whether what we were using, this 4 millimolar concentration, which is the standard concentration across the field, and we thought that something is wrong, because it is not possible the cells can revive from complete cell death if it is the truth. So we did concentration curve, and you can see that these 4 millimolar are still better than 8 millimolar, but you see the 8 millimolar, nobody can predict that these cells going to revive, but they will revive. Okay, so they revived. All of them, most of them. Here you will see all of them, there you see most of them, and 12 millimolar you will see none of them. So we found that this is a sublethal concentration which people were using, and it's actually wrong to use it, but it gives me another thought process to follow. So, and we found was that, okay, there is one more video. What we found was that cell death, what people use in the literature, is by these annexin FITC. If the membrane of the cell gets these annexin FITC positive, they take it as a cell death. But that's completely wrong. Until the nuclear membrane, the nucleus inside the cell, until this disrupts, there is no cell death. They can revive back. That's what we showed in the paper, and this could be a redefinition of proper redefinition of cell death. And you can see here that these cells revive back because they have not taken this stain into the nucleus. Once they take a stain inside the nucleus, because of the nuclear membrane disruption, they cannot revive back. So we defined it. We also defined how the cell organelles, different kinds of cell organelles we tested, check that inside the microscope. These are inside the cells. These are different organelles. You all know probably the tubulins, mitochondria, Golgi, and some complicated ones, autophagosome, endosome, lysosomes. We checked that at this stage, when the cells are going towards the death, they all get disrupted completely because these are floating cells. They are all getting disrupted. So from there, coming back to the normal life is very interesting for us in terms of cell biology, in terms of molecular biology, in terms of every biology process. It could be the possibles. So till now it was cell revival. Then we gone into the molecules. You want to understand what is happening, how these cells are reviving back. So this stage is mostly what I was showing. They come up, and these are all stages when they revive back to the normal. So we did the transcriptome to understand the level of gene inductions or mRNA induction, or to understand what is happening here, to be very easy on that. And what was very interesting will come from this heat map, that until the cell are dying, this was the C3 stage where the cells are in the supernatant and have all the properties of cell death. When the cell revive back, they revive back very interestingly. There are clusters of genes which comes up, and these are not 1 or 2 genes or 100 genes, or these are 2,000 genes. These are around similar 2,000 genes. So it's very well programmed to revive back the cells into a life. And when they revive back, I will not again take you what these cluster 3, cluster 2, cluster 1 are really. To be precise, what was happening is they are coming back to life, something like they know that what was then, they were like a embryonic developmental stages, they know that what they were initially. So when you see this cluster 3, the pathways which were activated, means the genes which were activated, is developmental signaling was getting activated. Almost everything you see, that all circadian rhythm, embryonic organ morphogenesis, cell development, muscular structure development. So these cells somehow are like, they have cleared what they were, and they are restarting their life. And you can see that what goes next is metabolism, nucleotide synthesis, protein synthesis. Everything was kicking in a very programmed fashion. So that's the reason we called it programmed cell revival. And if you Google that programmed cell revival, you can actually see more detail if you are a biologist and interested in it. Because of time, I may not go in that detail here. And for the biologist, what kind of pathway it was we found is, you can see all the pathways like Wnt signaling, NOT signaling, Hox signaling, DGF-beta signaling for the biologist. We found it, the first cluster 3 has all those things, and then you have metabolic pathways, you have inflammatory pathways, all these required for the revival of these cells. Okay, so again, this is quite a complicated. Just want to tell you that when the cells are reviving, all these gene signatures are not random. They take some kind of program. They go up, go down. Mostly they go up as they revive back. Okay, what's there? There was a transcription signature. We thought might be that's just not a transcription signature, but there will be a genomic signature also. So we start looking at the epigenetic level. So not going again in the detail, what we found it that at this stages, when the cells was in a completely apoptotic stage and when they are reviving back, the cells have lost their epigenetic informations. So this is the ataxic data, and you can see a lot of open chromatin at lot of genes, which means that when cells lost that, they lost their differentiated state, and they have partially acquired a de-differentiated state, which is more like a developmental cells or initial cells. So that's what chromatin opening data tells us, which leads to higher transcription at multiple loci. So that was the thing. This is all about LLOME being used to study how cell go to sublethal concentration of LLOME leads towards cell death. But if you actually give sublethal concentration, they can revive back. And when they revive back, they are like new kind of cells, which is more or less using or they slough off all the information, and they are reviving like they are in the coming to the new life. What was we thought of is that point of time that these all molecular pathways and looks like they are similar to what happens during wound healing, tissue regeneration, is and all embryonic growth. So they have a lot of similarity between these three processes. I can tell you there are tons of literature that wound healing, when the wound heals, they are also having similar pathways, which we are seeing even during the tissue regeneration. For example, you have tissue regeneration of in different models, tail regeneration or any kind of, these pathways are getting activated. Developmental pathways comes first. So we thought that let's check it out and whether this LLOME can actually cause some kind of interesting stuff in terms of wound healing, for example. First, we have done a lot of things now. This all work is filed for patent for India in 8 different countries now. So what we found was that this is name is changed, but this is LLOME. So what we found was that by the 3-day, this is a wound healing model where you can cause a wound on the mouse, and you can actually look for the different days, how fast it heals in multiple mouse. So one of the mouse we are showing, this is wounds which are made in the first day, and you can see that by the 3rd day, when the wounds are still fresh in the untreated mice, you have almost closed wounds with the LLOME treated topical treatment of the LLOME. We have another model with our collaborator where you can actually, it's called, you can injure the epithelia of eye with NaOH and scraping. And there also he saw the same thing, that if you treat with the LLOME, there was much faster recovery, much faster recovery. Within 1 or 2 days, the reepithelialization was taking place. There is another collaborator who was working on frog tadpoles tail regeneration model. There you can add LLOME directly into the water of these tail cut tadpoles, and when we put that, as you increase the concentration, there was much faster after the amputation, much faster tail growth. There is another collaborator who works in ACTRAC and also NBRC. So we try to understand whether the regeneration in C. elegans model, the axon regeneration in C. elegans models can be faster, and he found regrowth length as well as recovery index, which is these get recovered faster by when they dissect it and they cut this axon. First of all, regeneration was faster, and also the recovery of these C. elegans was faster. in another model, Drosophila, where they found that, very interesting, that the blood progenitor indexes, hematopoietic progenitor stem cells were increased in many folds in blood lymph glands when they are fed with the LLOME. So this is really interesting to us. We are trying to understand many things right now, and we are taking these different models, like this model also we are taking into the mouse model, which is now funded by ICMR by Center for Advanced Research grant. We are taking that model to understand, can we induce pluripotent stem cells faster. We are actually trying to understand whether we can actually heal the diabetic wound models faster. We are taking this to rabbit and probably to the human in this Center for Advanced Research grant. So with that, we start with the observation. We end up with multiple PCTs, multiple grants, and also beautiful basic work. These are all people who have collaborated, including IASc and multiple institutes all over the country. And this was the paper published last year. And this is my lab, and financial support with different agencies, highly thankful to them. Thank you very much.
One very quick question. Towards the beginning of your talk, you showed that as you increase the concentration, the number of cells reviving decreases. Later, what you show is as you increase the concentration, the speed of recovery increases. So is there a sort of opposite?
No. So the thing is, those are cells. These are whole body models. They are very resistant. They don't care much about that concentration. So cells are, because we are putting directly onto cell culture, that's what the dynamic is, that's what the concentration gradients were. But in human, so in the mouse, we found even 20 millimolar, we are giving from last 6 months, they are fine. So this different is because of different whole body models are very different from there. So there is no lethality by now we have seen with these concentration which we are using in the cell culture, to be precise.
Just one quick question. So have you looked whether in the context of tumors, whether these enhance tumor growth?
Very good question. So that's what we are looking for, what we feel, to be precise. In cancer cells, because I tell you what's going on right now in this field. This is, in the cancer cells, when we give all these anti-cancer drugs, our hypothesis is most of these getting into the stem cells is because of this process. Because you give so much of them, they are not going towards fully death, and they recover back with much potency, probably by programmed cell revival. So in the context of cancer, this could be a very bad process, because it allows these cancer cells with the resistance of equivalent to the stem cells to bounce back much faster with much probably potency as compared to the if they revive, if they don't die, or they revive without going this process, they revive or something like that. Yeah.
So 2 questions. First, when you look at different cell lines and different tissues, are the transcriptomic signatures at different time points similar, or do they vary?
So tissues we have not done. Now we are doing in wound healing model, which we're gonna go for time points. But in cell culture, it is absolutely same. We have done it with 2-3 different cell lines.
The second question, compared to an acute injury, when you look at, say, diabetic ulcer you are trying to think of, do you think the mechanism is going to be similar or?
So I think you are absolutely correct when you are seeing the cells versus you are seeing a model which is whole organism model, the many mechanism may kick in. And here also I am not claiming that I have found all the mechanisms. Because these cells who are going towards it, they will secrete a lot of EVs and lot of things which may actually call upon the immune cells. There will be a lot of things going on inside the organism. So in the inside organism, this mechanism may lead to, for example, macrophages, coming to macrophages inside this environment and clearing of the debris. And maybe the immune cells get activated. So inside the organism, maybe this mechanism may leading to many other kind of responses.
Good afternoon, sir. I was just wondering if, yesterday we had a talk by Professor Jagadish, and he had a shockwave way of healing wounds. I was just wondering if there is a way to apply shockwave first and then the pluripotent stem cells from LA1 can be added. In fact, you have some collaboration with the IASc. Maybe next time this is something that we could look at.
So Shockwave may be doing the similar. See, with this thing, what we have done is we have given a kind of mechanism to the world that may not be LLOME or lysosomal cell death driven. What we are exactly saying is, if cells are going towards, so Shockwave may cause similar kind of programmed cell revival, and they may induce similar kind of stemness. So that's maybe one way of doing the programmed cell revival for therapies.
Good afternoon, sir. I have a question, like in context of humans, do you think any cells specifically or like any types of tissues are susceptible to cell renewal, and why do you think so? And are there any biomarkers to detect this cell revival?
Very good question. The first one is, we mostly tested these cells in these things in fibroblast cells and epithelial cells. But if you go towards terminally differentiated cells, such as your immune cells, the U-turn is not possible by the day because we have tested it. So the terminally differentiated immune cells don't take a U-turn. So you have to have these fibroblast cells in your environment, in tissue environment, and epithelial cells, which is always the case. But terminally differentiated immune cells doesn't take a U-turn. What was your second question?
Are there any biomarkers to detect this cell renewal?
Biomarkers to check the cell. That's a good question. I don't know. We didn't try it.
One more. Like let's say the stress event that caused the cell to go to apoptosis or trigger the cell death, does it have like epigenetic memory? And if it does, like of the stress event, and when it gets like revived or gets under the process of renewal, right? So does it like the cell is becoming more aged? Does it age more fast, or like does it like have the memory of the stress conditions?
So what we found, it ages slow. In Drosophila model, what we have already checked, it's if you feed the larvae with the LLOME, the aging has, and this is patented also again, the aging is getting suppressed. Now we are going for animal model. Just I told you that we are checking that in animal model, and we have injecting them with that. So that's aging is slowed down actually.
So I'll ask the last question.
Sure, sir.
So what about in vivo? So in vivo cell revival, any incidents?
So we have checked in this paper to some extent. We cannot actually, we have done multiple FACS studies, multiple other ways of doing, taking the tissue from
without the injury I'm talking about, spontaneous.
Oh yeah, I'm sure that is happening.
No, but nobody has shown.
Means injury, this is injury lead. Wherever the injuries are, I'm sure this is happening.
No, injury is happening, but
normally our body all the time goes through the
programmed cell
injuries.
death.
For example, liver, we are injured by multiple dams. Our liver is one of the places where tons of dams are being accumulated and getting the injury. I'm not talking about the physical injuries. Our body normally responds, gives a lot of injuries.
Right.
And they're inducing the stemness will always,
So
stemness or
there maybe
redifferentiate.
revival is maybe happening,
Yes.
but you have to show that, right?
I can predict it.
Yeah, okay. But another curious thing is that whenever somebody is studying apoptosis, they do this dose-dependent things. So how was it missed earlier that lower doses are?
So, I don't know, but the
Because
12 millimolar, I don't think many people goes up to that, because they see everything by 4 millimolar. When they collect the cells, they go off. This
So
is
why
chance we didn't collect the cells.
Okay.
So
it's the
mostly
nature
4
of the cell.
millimolar people go, 8 millimolar may people go.
Okay.
There also revival is there, but 12 millimolar, I don't think someone has tried. It's just death in 5 minutes.
Okay. All right. So we'll stop here
Thank
and
you, sir.
thank Santosh for a very, very interesting